microbiome diversity is Primer-based methods. So you use PCR to amplify a specific chunk of DNA that exists on all of your microbial taxa, and then you can look directly at genetic distance from that amplified DNA. So it has some really great properties. The drawback for this approach is that you need to design your primers ahead of time. And so if you have specific, and you need to look at existing DNA sequences in order to do so. So essentially you may be very interested in things that are occurring in the microbiome, but if there's no representation of the DNA sequences of those organisms, you have no way ahead of time to design your primers to actually catch those organisms. And so you essentially have a bias towards like unknown unknowns in your sample set. So what I did instead, just as a first pass, is to randomly fragment the microbiome DNA with enzymes. And so this is a different set of biases because enzyme cut sites have their own biases, but again, it's a separate way to look at it. The drawback to this is you're not amplifying the same chunk of DNA, so you can't directly compare. You have to rely on publicly available, already identified DNA sequences in order to know what you have there. But the upside is that you don't have the primer bias, and you also know if you have a bunch of DNA sequences that you can't classify, that you need to do more work to figure out what's going on there. So first of all, I'm going to show you the bacterial results, and I'm showing these essentially as a sanity check for my method, because we know pretty well what's going on in the bacterial side of vertebrate microbiomes. And we find the 3 bacterial phyla that we really expect to find in the vertebrate microbiome. Those are the Bacteroidetes, the Firmicutes, and the Proteobacteria. And so it seems like this method is successfully identifying and amplifying gut microbiome DNA. And just as another look at how this method handles diversity of the microbiome, on the bottom here I have the number of total sequences I have, and then I have the number of new species identified by this method and the number of new genera as you increase number of species. And so you have roughly a 3-to-1 relationship in which for every 3 new chunks of DNA you look at, you get 1 new identified taxon in terms of species. And I'm only showing the lizards here in interest of just readability. Moving on to the eukaryotes, we see a very different scenario. So it's more of a 1-to-1 relationship, and the species accumulation is still going straight up as I run out of sequences. So clearly there's quite a lot of eukaryotic diversity here, and I'm probably not capturing all of it with this method. Also, there's a pretty large bias in terms of just the number of different eukaryotic sequences and the eukaryotic diversity that I'm getting from my 3 different hosts. But I do find 6 different phyla. 2 phyla up here of fungi, and then 4 different phyla, the Apicomplexa, the malaria parasite is probably the most famous example of this, roundworms, flatworms, and then cnidarians. These little myxozoans up here are nested within cnidaria, but they are very much gut parasites. They're drastically reduced to a couple dozen